Welcome to another episode of the MEGIN Spotlight series. Today we are delighted to have Dr. Lucia Amoruso, Ikerbasque Researcher at the BCBL in Spain as a guest. Dr. Amoruso leads the line of research focused on the identification of neurophysiological markers supporting language plasticity in brain tumor patients via means of MEG. Polina Timofeeva is a Ph.D. student working with Dr. Amoruso, conducting her thesis in the same field. In addition, BCBL's research activity aims to unravel the neurocognitive mechanisms involved in the acquisition, comprehension, and production of language, with special emphasis on bilingualism and multilingualism. Hi, Lucia and Polina. Thank you very much for joining us today.
Hi, how are you? It's a pleasure being here.
Thank you.
Hello. Thank you for having us here. We're very excited to share our research with you.
Thank you. We recently had an introductory call, and it was fascinating to hear about the incredible clinical support you provide brain tumor patients in collaboration with the neighboring Biocruces Hospital in Bilbao, utilizing MEG in the process. We'd like to find out a little bit more about this and about BCBL as well. So could you please tell our audience a little bit more about this about BCBL as a research and clinical institution, and what is the main focus in general? So Lucia, why don't you start with this question, please?
Okay, thank you. So BCBL stands for Basque Center on Cognition, Brain and Language. So it's a world-class multidisciplinary center that is doing research and training and knowledge transfer within the field of cognition and language, and with a special emphasis, with a special focus on bilingualism. So importantly, one of the main lines of research within this institution is focus on language and brain damage. And this is the line in which we currently work with Polina. So among BCBL facilities, there are many techniques. I mean, you have an MRI scanner, you have EEG systems, and of course you have the MEG system. So this wide range of techniques allows tracking different aspects of brain structure and function. And depending on the advantages of each of these techniques, you can ask different research questions, let's say. So for instance, given the high spatial resolution of the fMRI is great when the research question is focused on the localization of a given cognitive function. So which brain areas are involved in this process. However, hemodynamic responses are slow. So we have a scan, let's say every 2 seconds, and functions as language, that is the function that we mainly study, occurs at the sub-second time scale. So this needs to be examined with high temporal resolution techniques. And MEG meets this requirement as it can capture neuronal activity at the millisecond level. So it's offering like a new perspective to study language processing. So another important aspect that MEG is providing, unlike fMRI, it can capture the temporal dynamics of oscillations, so contained in the brain signal. Oscillatory activity is thought to play a key role in neural communication. Okay, so and also is thought to reflect distinct cognitive operations at different frequency bands. So you have alpha, theta, beta, and these provide a fine-grained characterization of the neurophysiological mechanisms supporting language and cognition. So there's a well-known metaphor from Bastiansen and Haggard. They say that trying to understand language function by relying exclusively, let's say, on the hemodynamic responses would be like trying to understand a piece of music by making an inventory of the instruments that constitute the orchestra. So in other words, language function is not just a collection of brain areas. And what we are missing in this description is like critical information about the melody played by each instrument and how they interact in real time. So this is the information, the critical information that I think that MEG can give us.
MEG is a vital modality at BCBL and used for research projects. Please could you explain how MEG is utilized at BCBL? Lucia, maybe you can answer that one first.
Yes, sure. Yes, as you were saying, at BCBL, there are multiple ongoing projects using MEG. Some of them are focused on more basic research aspects, and others involve this translational effort in bridging basic experimental research or knowledge with the clinical aspects of the research. So this latter line of research involves the study of language disorders, such as dyslexia or language deficits associated to neurodegenerative diseases, for instance, and of course, brain tumor patients. So this is the last one is the group in which we currently work with Polina alongside other great researchers, just to name a few, Ileana Quiñones, Xuan Geng, of course, Manuel Carreira, that is the director of the BCBL, and the neurosurgeons team in Bilbao, so Santiago Gil Robles and Inigo Pomposo. So briefly, in our clinical research, we use MEG to track longitudinal plasticity in patients harboring low-grade gliomas. So we are measuring brain activity before and after tumor resection. This type of tumors, low-grade gliomas, constitute a very interesting model to approach the study of brain plasticity, because actually they are not like lesions such as a stroke that occurs instantaneously and destroy the tissue, hindering successful reallocation of the brain functions. Low-grade gliomas infiltrate cortical and subcortical structures gradually. So they grow like 4 millimeters per year, let's say. So they give time to the brain to adapt and, let's say, to move the functions that were previously located in the lesion areas to healthy lesions, to healthy regions in the brain. So this obviously avoids severe cognitive or behavioral deficits. So when the patient comes to the BCBL, if you make like a classical exploration, neuropsychological exploration, there won't be a difference. I mean, it will behave like a neurotypical person. However, maybe if you use more fine-grained tests, you will find some cognitive deficits at that point. So basically, as I was saying, patients come to the BCBL. They are first recruited at the Cruces Hospital in Bilbao, that is a city next to San Sebastian, where the BCBL is located. So there is where they receive the diagnosis and they perform the surgery for tumor resection, so the awake craniotomy. So the patients come to the BCBL before the surgery, and they perform an fMRI and an MEG session. And the information that we get at this stage is used to perform a report for the neurosurgeons to jointly plan the surgery strategy in a very personalized way. So selecting those tasks that better suit to the patient profile, linguistic profile, and lesion location. So for instance, I don't know, if the tumor is compromising language related areas, we run linguistic tasks. So naming, reading, or listening to sentences and saying if they are correct or incorrect. So we use this to map language function. And of course, if the patient is bilingual, we run them in the two languages the patient speaks. So then during the surgery, we select those tasks that are better suited for that patient. And in combination with direct electrical stimulation, we map the language during the surgery. So the patient is awakened, and we run the tasks, And while we're running the task, the neurosurgeons stimulate these brain areas surrounding the tumor. If the patient performs an error, a spatial rest, anomia, semantic paraphasia, these are different type of errors, it means that that area is functional, I mean, is housed in function, so we cannot remove it, so we will preserve it, okay? Mm-hmm. So this procedure allows maximizing tumor resection while preserving cognitive integrity, language, and motricity. So then finally, after the surgery, patients come to the BCBL 3 months and 6 months after the surgery. And they run the follow-up session. In that session, they run exactly the same tasks that during the presurgical stage.
So why are the findings and data provided to neurosurgeons so essential when dealing with brain tumor patients? Polina, maybe you can answer that one.
Yes. So at the moment, the most popular approach to map preoperative function is using fMRI, and indeed that has been used for decades, and it's been advancing our understanding of brain and the function for that long. But unfortunately, fMRI measures hemodynamic response, as Lucia mentioned before, which is first, not a direct measure of brain activity. Second, it cannot allow us to tap into the processes that happen at the millisecond scale. And third, fMRI signals are affected by tumoral effects such as edema, for example, and changes in blood supply, leading to inaccurate functional mapping. So this is where MEG comes into play, because it has a great temporal resolution and also spectral resolution. Also, it's not as affected by the tumoral effects. So it allows us to study brain function and plasticity at the millisecond scale that we want to see when we study language, for example. So as an example, by exploring how activity unfolds in time in the brain, we can identify whether patients use the classical ventral routes, for example, involved in reading, or if due to the compensatory mechanism due to a growing tumor, they switch to a more alternative dorsal routes. So these responses normally happen 170 milliseconds, around 170 stimulus. So for example, continue with the reading task, with the reading example, like they see a word on the screen and they have to read the word. So that first response happens around 170 milliseconds. And this is why the temporal resolution of MEG becomes critical. So all of these things make MEG a great tool to assess oscillatory dynamics and network organization in both neurotypical population and also in brain tumor patients. So in a summary, all of these factors make MEG an essential part of the report that we provide to neurosurgeons before the surgery.
Polina, in your research work, the protocols that are run in MEG and fMRI pre-surgery, depending on each individual patient, they result in different outcomes for each patient during surgery. Explain to us what impact these protocols have for the clinician and how the results are utilized in surgery.
So, as Lucia said at the beginning, the center, the BCBL, is located in the Basque country, which is a bilingual area in Spain. And most of the population speaks Spanish and Basque at a different degree. So I would say that most of the patients that we get are actually bilinguals. So the different tumor locations. This implies that we have to create a surgical strategy that is adapted to each patient to optimize the benefit to risk ratio of the intervention in a personalized fashion. So in the report that we sent to the neurosurgeons, we offer a detailed description of the brain networks that become activated in each particular task. So we also provide the timing of the effects that MEG is perfectly capable of providing, their lateralization, and importantly, if the implemented task that we run with the patients allow mapping brain activity surrounding the tumor. So those tasks that do allow, do have the sensitivity, we further use in the during the surgery, during the awake surgery. So the patient is awakened during the surgery, and they go through these tasks. So as I said, the task change from patient to patient based on many different factors, as like personal information, like the profile of the participant and the sensitivity. So as one example is continue with the bilinguals, well, based on the bilingual population that we have here, the one example, and actually a part of my PhD thesis as well, is the bilingual patients. They have to deal with language control. So during, like in your daily life, they have to alternate between languages while avoiding interference between them. So in those patients, the main focus for us would be to preserve both of the languages of the participant, and also to preserve the ability to actually switch between them.
Lucia, you also studied the same patients post-surgery. What are the findings, and could you tell us about a specific case to illustrate how pre-post-surgery MEG protocols have been vital for the patient?
Okay. Yeah, actually, while the information from the pre-surgical stage is mainly used for clinical purposes, so we use this information to make the report for the neurosurgeons and to plan the surgery strategy. The post-surgery information is used more with a research aim, let's say. So specifically what we do is to compare post and pre-MEG responses. So we try to better understand with these neuroplasticity mechanisms that are put in place. And this longitudinal approach also allows us to evaluate whether the patient recovers in a satisfactory manner after the tumor is resected, and if the functional reshaping triggered by the surgery was successful. So, for instance, we have recently published a paper in Cancer's Journal conducted on a group of Spanish Basque bilingual patients. So, briefly, what we find is that monolingual and bilingual individuals, they put in place the same neuroplasticity mechanisms to deal with language reorganization and preservation. So they recruit ipsilational regions of the brain that are healthy. And also, when this is not enough, they recruit contralational areas from the other hemisphere, typically the right one. So we also find that language rearrangement in the dominant and the non-dominant language, I mean, that this plasticity is occurring in both. We say the L1 and the L2. So what underscores this is that we need to map all the languages during the surgery, all the languages that the patient speaks in order to preserve them. And we also observe that this reorganization that is occurring after the surgery is different for each of these languages. So what we find is that bilinguals have common but also dedicated areas for their languages. So another finding is that it's not just the language network which is involved in this functional rearrangement, but also areas associated with executive control, for instance. So this suggests that the reorganization is more global, more holistic, let's say, and it's not circumscribed to the language network itself.
Thank you. Polina, explain to us what is the sort of long-term goal of this project for you?
So we do have a lot of data. So I think that in the long term, we plan to use this information to predict patients' recovery in both bilingual and monolingual populations, while trying also to unveil the main factors influencing this phenomenon. So in other words, our future goal is to predict personalized recovery trajectories, combining MEG, fMRI, behavioral data that we have, and apply machine learning algorithms on that. Unfortunately, at the moment, we do not have that much data, that many patients. And so we are now at the stage of collecting more data as we go.
Oh, that's great, thank you. So a bit of a different question, I guess. Why is working with MEG a rewarding career path for you? So, you know, what advice would you offer other researchers looking to enter the field? Maybe Lucia, you first, and then I'll ask you, Polina, as well.
Okay, I truly encourage researchers that have the opportunity to enter into the MEG field to do it. I am sure they won't regret it. In my personal case, I just love MEG. This technique is wonderful because apart from providing a direct measure of neuronal activity, it allows you to answer research questions about the timing. So when a given process is occurring actually in the brain, and also the brain rhythms involved in these processes, so, such as, for instance, the case of language plasticity. And all this information would remain undetected using traditional fMRI approaches. So MEG signal is very, very rich, and when analyzed with the proper methods, can provide accurate estimations of different aspects of brain function.
And Polina, what about you then?
So I agree with Lucia. I think it's a fascinating field, and as a young researcher, I also encourage others to give it a try because I find it fascinating. So I have a background in control engineering, so I find it fascinating that even though the field has been around for decades, the analysis has been evolving very rapidly over the past, I don't know, 10 years, for example. And now you can use different techniques, different analysis. You can create your own analysis, the pipelines or new methods, and apply signal processing techniques as well. So either you want to work in, like, if you want to create new methods, you can work with MEG. If you want to just study how our brain works, you can also work with MEG. And I think it's great for everybody.
And I guess in your case, you can see the sort of broader picture with all the other modalities that you're using together as well. So that's helpful too then, isn't it? Thank you both for speaking with us today. It's been really great. We have really enjoyed hearing about BCBL's projects and how MEG is being utilized in conjunction with brain tumor patients. So thank you so much again for your time and for coming on our episode today. Thanks.
Thank you for having us.